All right, so maybe I get started. First, I would like to thank the organizers to bring us all here, and so to make our life as nice as we have it here. So I really appreciate that. I also want to thank all the other speakers so far, and I guess also for the future ones, so for their illuminating and exciting talks, and also so to give me some duties to cover. That's also very nice. But I will continue this game, and everything that I cannot cover, I will just put on the following speakers. Okay. Um, yeah, since I do only have this 1 hour and a bit to brainwash you, I had to downscale a little bit the aim or the scope of my talk, so that's why you won't get the theory of everything, but I scaled it down, and I will talk to you about calcium dynamics as a mediator between energy, metabolism, and cell fate. So, but why did I end up here, or how did I end up here? So, well, actually, there was a forming moment actually in Corsica a little bit less than 30 years ago, where after a high school celebration, I went by myself in my backpack to find myself, or to re-find myself after all this high school things, and was hiking through Corsica, and I came to the conclusion so that I either want to become a rock star or a physicist. And then back then it was actually a good way to go to Berlin. And why did I want to become, well, a rock star is obvious, but a physicist, so a little bit maybe afterwards driven by this famous Rutherford saying, so that all science is either physics or STEM collection, which is totally arrogant, and also, if you take it too serious, not well thought through, but since he won a Nobel Prize, right? But as you can see, Nobel Prize for chemistry, right? So, but on the other hand, you know, I think that's actually related to what I'm doing, because I think that physics is a little bit more like a thinking school, right? So, and then it's about understanding things, putting things in relation, or as Matthias phrased it, identifying the patterns or something like that, identifying correlations, so causality. And in this sense, I relate to Rutherford saying so. And then I had another decisive moment when I realized so that I'm either too stupid for string theory, or that string theory is at least not made for me. And then I diversified myself and went into theoretical biology. So I was lucky that back then there was kind of one of the first schools of theoretical biology in Berlin, and then they caught me. So I realized, okay, I can do something useful with the math that I already knew, and can identify patterns that I can try to explain. I went then on, did my PhD in biophysics, where I then also started to do experiments, and afterwards, so it was the big times of systems biology, and then now I actually ended up in Luxembourg, where I do systems biomedicine, where my lab is doing both, so experiments as well as theoretical and computational approaches, where we try to integrate across the different scales of life, and mainly of the brain, for a better understanding of the organism, and hopefully maybe we have also a little bit of an impact to diseases. Okay, that's me. Any other questions, or we keep them later for a beer? My shoe size is 42 or 43. Okay. Okay. And another decisive moment for another person. So this is my daughter, and she will get married next weekend. So, and that's why I have to already leave on Wednesday. So it's not that I'm then saturated or do not like the place, but otherwise I will be killed by her. Okay, so that's why my apologies that I have to leave on Wednesday. Mainly probably pity for myself. All right. There was, in Matthias' talk, there was a claim for numbers, yeah. So, and since it's also a little bit a driver why I ended up in the field, is a complexity, yeah. So biological systems are really complex, and if you like complexity, then it's a good way to go. And I think that's probably a theme that we share, or that share many of us. The human DNA has this 3 billion base pairs, which is approximately 1.5 gigabytes, and we all carry about 3 million mutations. So Luckily, most of us do not have very severe mutations, but this is what is making us the individuum that we are. And if you copy this number with the number of cells that you have in your body, then they on a back of the envelope calculation. So actually, if you would then save that on an external hard drive, and you staple them up, so then you would be 10 kilometer high, so even larger than the Eiffel Tower, to relate to other back of the envelope calculations. So we have this 20,000 genes, and that are coding for proteins. And these proteins do a lot of interaction, so that we have in the human about 200,000 protein interactions. If we zoom into a cell, so typically we have one DNA, then we have 10 to the power of 5 mRNA molecules, and around 10 to the power of 8 proteins. Of course, there are cells that are larger and they have more, and cells that have less. And then we have this other molecules, so Matthias told you, so right, so 70% of a cell is still water molecules, but then we have also the small molecules, so metabolites, that we will talk about later on. And a fun fact is so that the human body has also at least the same amount of bacterial cells than human cells. So there are other estimates that say it's even 100 times more. So the microbiome research is there still conflicting. What's your favorite number, Anupam? Okay, so you see, you know the field that he's working in. Okay, that's about the numbers. Anybody knows about the central dogma about molecular biology? Or does anybody not know the central dogma about biology? Okay, everybody knows. So then just to give you some other numbers, right? So we have here 2,000 transcription factors in humans that do the regulation on the mRNA level. So, and mRNA is, well, DNA should be rather stable. mRNA is more flexible. So that's why it's also the level where a lot of regulation is happening. Yeah? So the gene regulatory networks are very often described on the mRNA level, where this 10 copies per cell is now also per gene. Yeah? So, and then that's the first part, the transcription, and then the second part is the translation part, where then the messenger RNA is translated into the proteins that have again then a higher lifetime time than the mRNA. Given my talk here, so then actually central dogma of molecular biology from the 90s. So we now so that metabolites play a major important role in all of this one, where then the specific category of proteins, the enzymes, do a lot of this metabolic regulation. Okay, everybody knows the ohm word as well. So Carol gave me the duty to speak about the ohms. Ohm is clear? Okay, the genome, so the ohm means the completeness of something in biology, right? So meaning the genome is all the genetic information, the transcriptome is all the information that we have on the RNA level, on the transcriptome, and the proteome is everything that we have on the protein level. And then classical dogma of molecular biology, so straightforward, but of course biology lives from the feedbacks. So, and the closer we look, the more feedbacks there are, so meaning that in the environment, so we take our metabolites from, that can have then strong regulatory effects. So polymerases, ribosomes, enzymes, so for those who are not from biology, I think that should be your favorite proteins, because they keep you going. Coming back to my rockstar, so that rockstar ambitions, so I can also put that a little bit into the musical framework, right? So the DNA is like the piano, so giving you the toolbox that and describing what is possible. So on the transcriptome is then the transcript that is actually how it should be played. The proteome is then the implementation of it, and then the metabolome can be maybe seen as the environment that is then having a feedback back to the actually protein level. So how do we measure all these things? So just very briefly, so I think the revolution in biology, or what we now know in biology, is really about the high throughput methodology, so that we can now measure large part of this ohm. Yeah, so we still do not measure everything, but we can now measure with some reasonable price a lot of things. So the sequencing revolution allows us to now get the DNA and the mRNA information, and the other major technique in biology is mass spec, right? So meaning where we can measure abundance of proteins and metabolites. And so we are still pushing the boundaries there, so we try to increase sensitivity, and very much related to that, also try to understand how we get better signals out of it in terms of data analysis and data handling. Is this enough for the omics, Carol? Do you check me off? Yes? Okay, good. Then I come to my actual outline, where I will start a little bit with my big picture of life, and then we will go a little bit into the multi-scale regulation of energy metabolism, where, well, I will start with the calcium dynamics that is strongly related to information, so also making link to theory. Then calcium regulates mitochondrial metabolism, so meaning here we have the energy, and if I still have time, I will also talk about adaptation, differentiation, dynamics, which I consider as a nano-evolution. Yeah? So, and then I cover the three buzzwords of this workshop, where I had to swap for educational reasons the order. So I start with information, energy, and evolution, and then let's see if we can come to some kind of conclusion. Okay, the big picture. So luckily for me and Matthias, so scientific language is English, so that's why it's more like two great minds alike. In German, the saying is two idiots, one thought. So, but maybe since I relate that in terms of Schrödinger, so maybe the English is right here, and it's more two great minds like. So, being really arrogant. But anyway. So what is life? So life is a black box that we try to understand, and so I'm fascinated how this molecular soup is organized into something that biologists call the phenotype, which is then more robust, structured, and ordered than the molecular soup. And one of the first person that has looked into that one was actually Jan Swammerdam. Has anybody heard from him about it before? So he was a Dutch biologist in the 17th century who was fascinated by morphogenesis. So that's why he did a lot of stem collection, as Rutherford would say, so, and was trying to make sense. So how do the different species relate to each other? How does a butterfly develop, and so on? But he already had some interesting thoughts in terms of how you can try to make sense out of a stem collection. Okay, so what is needed to go from the molecular soup to this phenotype? So we have to bring order, right? And then we are with the Schrödinger, famous Schrödinger quote. So the order from disorder, which is then organized by complex out of equilibrium self-organization, and with the aim maybe for some robust information conversation and replication. And to describe that, we have another old, not so old guy here. So Alan Turing, who actually then used reaction diffusion equations to describe embryogenesis. So meaning we have here three people, and Alan Turing, well, mainly known for his machine, the Turing machine, but for biology, probably his work on embryogenesis might have been more important. Okay, so for those who didn't guess, so I briefly stay on the Schrödinger picture, where probably all of you know this idea, so that to generate order, we have to need to do some entropy export. And why are we able to do that is because we are on the Earth that is radiating the same heat that it's getting from the sun at a different temperature. Yeah? So now, well, with climate change, we obviously are not totally there in equilibrium, but anyway, so on a rough scale, we have there a negative entropy result that allows us to generate some order. And then you can scale this a little bit down to Schrödinger's cow, as I call it, and maybe Schrödinger's cow is also more important than Schrödinger's cat for biology, where the argument is a little bit the same. So what is the cow doing? So it's eating grass. What is grass? Grass consists of starch, and starch are long chains of glucose molecules. Huh? So long storage of energy. The cow is doing metabolism of it, is cutting these down to some shit. Now, if we do again some hand waving entropy calculation, so we see actually that we have there in the beginning a low entropy, afterwards higher entropy, so meaning that the cow, or the entropy of the cow, can be negative to produce some structure. So yeah, so one could now also maybe ask if the Einstein relation is more important than general relativity, but that we skip maybe for a discussion. Okay, so nice picture. Is there any relevance to that? Can we understand this a little bit better? And actually, there are some enzymes that do something similar. So there is this disproportionating enzymes, DPE called. So they are in plants, and what they are doing is that they take glycogen, so also something that Matthias already introduced, which is a subclass of starch, if you want to mean. So it's a linear chain of glucose molecules, and what these enzymes are doing is that they take two of these molecules and they shuffle around glucose units from one molecule to the other one. Yeah? So meaning they cutting off one bond here and transferring it to another one. So and then the specific enzyme here is able to do this with one glucose unit, two glucose units, or three glucose units, meaning so that in one reaction step, one of these enzymes, starting with homogeneous solution where we have all change of length 4, in one reaction step it can either generate this pair of 1 and 7, or 2 and 5, or 3 and 4. Yeah? Is that clear? So just by mixing, and since it's opening and closing the same kind of bond, so there is no enthalpy production. Yeah? So it's from this sense neutral, so that's why it's only driven by entropy. So where for a closed system, can you still see below here? So meaning for a closed system, we can then describe that so that we have mass conservation, and the number of bonds will also be preserved. Yeah? So now in this way, we can directly write our entropy here. So Alexander has put this already on the slides, and Thierry as well. So where now the entropy of the system is given by the length distribution, by the entropy of the length distribution here. So x log x, and the sum over all lengths. And then for the steady state, what do we expect? Maximum entropy, right? We have a closed system, so we would expect maximum entropy, so meaning we would expect that we get this kind of distribution where you can actually do the math with the specific constraints that we have here in the system, where we then see that the equilibrium, no, I spoiled myself. Can go back, please. I agree, Matthias, this is confusing. Where we get from the maximum entropy principle, so we get this solution for the length distribution, and so the parameter here is only dependent on the number of initial bonds that we have in the system. So is this actually true? So and now people may ask, why was Matthias complaining? Yes? What defines? Yes. So in the end, yes. So it's mixing. The entropy. So what do you mean? The enthalpies, or no? They are the same. They are the same. In the first reaction step, right? So if you start with homogeneous solution where we only have four lengths of 4. So then in one reaction step, this enzyme can generate this one. So then in the next reaction step, it can take now any of those as well and transfer again 1, 2, or 3 units from one to another. Sorry? Yes, this can go because you do not need energy here because you open closing anyway the same bounds, so it's energetically neutral. Okay, so then if you put that in a test tube, what do we see actually is that the final distribution, the equilibrium distribution, only depends on the number of bonds that are in the system, right? So as we expected. So it doesn't matter if you put there 100% of chain length 3 or if we have a 50-50 mix of chain lengths 2 and 4. So the equilibrium distribution is the same, the steady state distribution. The same is true if we have, if we do this for 4, so either with a homogeneous mixture or a 50-50 mixture, as well as for dp5. And you see so that the longer the number of bonds or the higher the number of bonds becomes, so the more flat the distribution becomes. And here you see actually this, the lines are the theoretical predictions and the dots are measurements, and everything looks very nice, but this is in a test tube, right? Here it's not cells, but it's enzyme in a test tube. Anyway, rather remarkable that theory and experiments fit so nicely that you cannot even see sometimes the error bars. The slope here, so this is this beta parameter here, right? So the slope, so it was a log plot, so that's why this is the distribution here. So, and the slope is determined only by the number of bonds that you have in the system. Okay, so then you can think about another important enzyme, so the alpha-glucan phosphorylase. So what this enzyme is doing is that it's acting on this glycogen molecules and is putting phosphate left or right. And why is this interesting? Because when it's putting here, if you end up with a single glucose unit with a phosphate, we have glucose 6-phosphate, so that is then entering directly into metabolism. And so you can do the same calculations as before, and you see the steady state distribution for 2 different initial values. And again, so fits more or less perfectly in line, meaning that we really have here an enzymatic system that is driven by the mixing entropy. So does this have any implication? So that was then in the review process that we had to show that that if you now consider a system where you have maltose input, so permanent input of a chain length of 2, so and then you have this disproportionating enzyme that is building the diversification, and afterwards you have a phosphorylase that is then pulling out the glucose 1,6-phosphate for metabolism. So this guy here is then used for growth or other cellular processes, and you can compare that with another enzyme, the maltose phosphorylase, that is not having this soluble heterogeneous glycogen buffer, but is directly getting the glucose 1-phosphate for metabolism. And if you do then the in silico experiment that you have in fluctuating input of maltose, then this system here where we have the diversification of this pool, so then these fluctuations are damped out, and specifically at the end of any input, right? So we can still then eat up the storage, whereas the maltose phosphorylase directly runs into energy deprivation. And in terms of Matthias' comment about a flux sensor, or today Terry mentioned input-output relation, if you have the maltose, if you look now into the maltose influx and the glycolysis activity, so if we have this buffer, we generate some stable and more robust conditions, whereas for this direct action of the maltose phosphorylase, we have going back and forth directly with the input. So then depending on the system, what you want to do, so one or the other might be more beneficial. Okay, so then everybody knows now what CRN stands for. Yeah, so chemical reaction network approach. So that is then work here from Massimiliano's group that we're then developing a framework to such a scenario. You can write the entropy production rate in this form, and then you see again what Fra told you. So you can split this up into internal species and the chemostats, where now for the easy case that we also could that we also solved with the maximum entropy principle. So this reduces then, or you can then get this form for the steady state concentration with the more or less same constraints as before, right? So mass conservation and the conservation of bonds, and you end up with a very identical prediction of the steady state resolution distribution. Okay, so far we haven't gained anything, but what we can do now, or not we, but they can do, is that you can now actually play around and go for non-equilibrium in the chemostats. So here is now the scenario explicitly written down for 3 chemostats, where there's one chemostat here, so we have extraction, so it's fitted to 1, and we have inputs at these positions of this chain length, and with that you can actually now predict the steady state distributions for the system without any simulations. So just as another motivation why CRN is actually a useful approach and can be used to understand and predict behavior of non-equilibrium systems. Okay, so meaning, bottom line is, life is kind of a smart way to use energy, so to organize itself as a self-organized system. Therefore, the system has to be open. We have as an input, we have cell signaling and metabolism. Then the cell is somehow using this input in an adaptive manner to adapt to the environment, and is then signaling back into the environment how it feels and what it wants to do. Describable by a complex reaction diffusion system, right? So where the magic of life lies into this plethora of reactions that we observe in a biological system. And I call this then sometimes also cellular evolution or nano evolution, to just bring 2 other old guys on the slides here, which are probably then related to this general procedure of induction and selection. Okay, so the smart way to use energy and repetitio matrix scientiae is we have this 3 main energy pathways. So glycolysis, so taking glucose, running through pyruvate, and then the pyruvate is a substrate for mitochondrial metabolism. Glycolysis, low or lower gain of ATP per mole of glucose. Mitochondria, higher gain, so but for sure smaller than 36, as you learned from Matthias. We have then also the pentose phosphate pathways that is going out here, and we have something else that I will briefly touch later. Also, is that pyruvate can also go into lactates when Matthias is running in the morning, or I'm swimming in the evening. So where we have then the lactate dehydrogenase that can shuffle around and is actually in the brain also an important energy moderator. Okay, so smart means maybe regulation, and then Francesco already spoiled that, so that if we look into the TCA cycle, that there are 2 enzymes that are strongly regulated also by calcium, so the isocytoid dehydrogenase and the alpha-ketoglutarate dehydrogenase are regulated by calcium as well as by the ATP level, meaning so there is a feedback mechanism how the cell can sense where do I stand with my energy, do I have to adapt something. So the PDH, so meaning the other way around here from the pyruvates into acetyl-CoA, is also calcium regulated and also depending on ATP, which are the main key regulator of energy metabolism. One way to go into the TCA cycle is obviously the glucose, but I think either Carol or Matthias mentioned that there are also other pathways how you can feed. So either you go from glutamine into the alpha-ketoglutarate, or you can use beta oxidation to fuel your TCA cycle and to generate ATP. Okay, so now we have seen calcium is regulating that, and that's why you will see that calcium is also often named life and death signal, because it's a very versatile second messenger that brings external signals, so in terms of hormones, for instance, including but also ATP, for instance, external signals into the cell, so by gene-protein coupled receptors that are then doing generating IP3. The IP3 is diffusing into the cell, and then there are receptors on the endoplasmic reticulum that will open in dependence on the IP3 concentration and the calcium concentration, and then flooding the cytosol with calcium. And from the cytosol, calcium is then pumped back into the endoplasmic reticulum and into the extracellular space by calcium ATPases, so meaning pumps that use ATP to push calcium against the ion gradient. So calcium is well, I don't know what the right word is. Well, people still believe, or and we have some evidences that calcium is frequency encoded, meaning so that changing the environment can change the frequency. So that's something that Michael Berridge established. He never got the Nobel Prize for it, so he's still, well, now he's dead, but he was suffering about this a lot, and maybe it's a little bit unfair, because calcium is involved in so many processes that it's got its name about the life and death signal. So it starts at fertilization, so when the sperm is entering the egg, so then there's a calcium wave running through the egg to prevent further fertilization. So then we would have, if this wouldn't be the case, there would be many more, well, not only twins, but what's the English word for tenth twins, whatever. After fertilization, so then there is a lot of regulation on the transcriptional level, also triggered by calcium. As we have already seen, mitochondrial activity is controlled by calcium. So what is another very important part where what calcium controls? Pardon me? Yeah, cardiac, and how? Yeah. No, so it's activating molecular motors, right? So meaning if you contract your muscle, so that's calcium, because the calcium is then actually pulling or activating the molecular motors to pull together your actin filaments. So and that's why cardiac was right. So because for those who closely looked, they sorry, I didn't hear you. So yeah, and then at the end of life of a cell, so also apoptosis is driven by calcium signals. So that's why it's called the life and death signal of a cell. So, and what I will talk today is, so more details are about mitochondrial activation by calcium. So there was this landmark Cell paper from Andrew Thomas in 1995. What you can see here is, so imaging the calcium in the cell. So then you see that such a calcium spike is followed by an NADPH signal, so which is related, so for those who remember Carol's lecture, linked to mitochondrial activity. And you see again, so if you spike again, then in the calcium concentration, you push always mitochondria. We still do not totally understand how and why and what, so, but I think I hope that I will convince you so that we got a little bit smarter on this front. And the other part, if I still have some time, is the other important experiment, another important experiment by Dolmetsch and co-workers in 1999. So where they actually used T cells, yeah, remember? So we were talking about T cells, and they were now forcing these T cells to spike with different periods, and were then looking into gene expression. Yeah? So Thierry mentioned today already NF-kappa B, yeah? So an important signal in T cells, and you see the faster the cells spike, the more of this mRNA is produced. So then NFAT, so it's the nuclear factor of activated T cells, also increasing the faster the cells to spike. So meaning there is really a strong regulation, and not only in T cells, also in other cells, but it's kind of the bridge between somehow energy metabolism and cell adaptation. Okay, so but how does this work, right? So there are calcium oscillations, or they're called oscillations. What do we need for oscillations? You're a physicist. A spring, okay. So that's good. Okay, so but how do you bring a spring into the cell, or what is the major feature of a spring? Potential energy, yeah. So a little bit more on the abstract level. Yes, feedback. Yes, thank you. So you need a feedback, right? So and that's actually in the spring, because there you have the feedback. So from in the end, from gravity or from your force, right? So the more you have potential energy, so yeah, okay. And then negative feedback is typically the one that is generating stable, more stable oscillation. So in complex networks, you can also generate under some artificial architectures. You can also use feed-forward feedbacks, but as a rule of thumb, so negative feedback is a good choice to generate oscillations. So another requirement for a continuous system to show oscillations. Huh? Pardon? Yeah, okay, but now we are already advanced. So I'm still on the easy peasy level. So dissipation, we forget about this. Just from dynamical system theory. Different timescales, not necessarily, but yeah. So it's also an interesting thing. So I think much more simple. Probably it's too much into your Poincaré-Bennigsen theorem. So trajectories cannot cross, right? So in the phase space, meaning, and for that one you need at least 2 dimensions. Huh? So otherwise, in 1 dimension to go back and forth without crossing the trajectory will be hard. Okay, so and with so this is a minimal requirements that we have, and then how can we do that for calcium oscillations? So the first very easy way would be to have an equation for the calcium in the cytosol and one in the endoplasmic reticulum, right? So and that was the first model that was put out there by Meyer in '89 and Goldbeder and Schmidt in the '90s, and this model works because it's always repeating, emptying, and refilling from the endoplasmic reticulum. So here you see the cytosolic calcium concentration peaks and the endoplasmic reticulum falls, and then it's going back and forth. So a potential explanation, but then actually the calcium is highly buffered and so on, so that's why there were some doubts if this is actually really true. And then there are experiments that show that even if the endoplasmic reticulum is so rich on calcium that you cannot have depletion, so doesn't really work. So another feedback mechanism, if you would be God, where would you put another feedback mechanism? One way could be on this level here, right? So where the input signal is dampened. So you get an activation, and then if calcium comes up, it can shut this down. Actually, for some cells, this is true. So, but for other cells like T cells, for instance, again, always the T cells, it's not true. There's not this feedback mechanism, but still cells do oscillate. Yeah? So, which level of regulation do we have left? Here, right? The channel itself. So, and that's then actually the thing that I will follow up a little bit more, where from these three different alternatives, so this one is probably the most robust, and well, because if you get rid of this feedback, you get rid of the channel, and if you get rid of the channel, you do not have any calcium flux anymore out of the endoplasmic reticulum. This said, all models are wrong. Some are useful. I hope that I can convince you so that we can do some useful things with some of these models. Okay, here you see now such a channel by a cryo-electron microscopy picture. So we look on the bottom. In the middle we have the pore. So if the channel is opening, so then the pore opens and calcium can go through this pore. And and the channel has 4 of these subunits. And each subunit has 3 different binding sites, or 3 different kind of binding sites. One for IP3 that is activating, sensitizing the subunit. One for calcium that activates then the subunit, and the dominant inhibiting binding site for calcium. Yeah? So, and now if at least 3 of these 4 subunits are open, so we learned this from patch clamp experiments, the pore opens. So, and the feedback is now realized so by the different affinities for these 2 different calcium binding sites, where the binding affinity for the activating site is lower than for the inhibiting binding site, meaning that if we have low or no calcium, so then the channel won't do anything because it's not activated. Then for intermediate calcium concentrations, the probability that this binding site is occupied is larger than this one is occupied, so meaning we get an increase in the open probability, and for even higher calcium concentrations, then the dominant inhibiting calcium binding site will be occupied and will get then inhibited ion channel. And that's how calcium flux is stopped. And then, so that's then something that is called, again, wrong button, calcium-induced calcium release, because if there's a little bit of calcium, you have a self-amplification mechanism that is then leading into saturation and inhibition. Okay, so then how does this look in a cell? So here you see a picture of a HeLa cell, and in this HeLa cell, the endoplasmic reticulum is stained, and you see it's a tubular network that's spreading through the whole cell. You see here the nucleus. If we then stain for these channels, the IP3Rs, then you see that they are not homogeneously distributed, but they form these channel clusters. Yeah? So, and in such a channel cluster, there are then typically between 5 and 15 of these channels, again shown here by cryo-electron microscopy, where each of these blobs here is one of those channels, and here you have actually a large cluster. And if you now take all this into consideration, the question comes a little bit, how does this spatial arrangement of the cell is acting on the dynamics of the cell? And since I'm obsessed with try to make some logics and some understanding, the question can be rephrased so that on the smaller scale, the molecular level, right? So we have this we have molecular fluctuations. So just by the inherent binding, unbinding of these molecules to the channel, activating and non-activating, and then you can see here on this very fast time scale of 200 milliseconds, we see a lot of opening, closing of the channel. So with a large variability in the opening times. Since these channels live together in these clusters, they do interact on the cluster level, and then they are generating something that is called puffs. So kind of collective opening of channel clusters. So here you see such localized events from simulations, and you see here so that based on the stimulations, so you see actually so that we can increase somehow the frequency, or at least the average frequency. And we get kind of such an exponential distribution for the opening times. And then on the larger scale, or the larger scale of the cell, so then is another important experiment by the Parker group, so by Martin co-workers. And what they have done here is so that they were screening the same line of a cell over time. So we have here the time, and then the microscope was always scanning the same line. At the white line, so IP3 was given to the cell, and then you first see that nothing really happens. Then you see such a localized event, yeah? So which corresponds to the opening of an individual channel cluster, yeah? So such a puff. But this puff alone doesn't do anything. Only if several of these openings come together, you then set off a wave. Yeah? So and then this wave corresponds to such a calcium spike in the cytosol. So and so any guesses already here? So if we from the name puff to wave transition by nucleation, what would you expect? Is nucleation deterministic? Spoiler, no, right? So there is a random part in the question that I will now first show you is how do these molecular fluctuations on the channel level are entering on the cellular level. Okay, another thing is obviously if we need this nucleation phenomenon here to bring these release events together, we have actually then a dependency on the spatial coupling. Yeah? So meaning if this one channel cluster is opening but the other is not recognizing it, so it won't get activated. So that's why there's a dependency on the communication, the spatial coupling between these release sites to generate a coherent cell-wide signal. Yes. Because you change here the stimulation. Yeah, so. Okay, so we then went and measured a lot of cells, individual cells of different cell types. So here you see an astrocyte, so that was just put under a microscope, and then you measure the cytosolic calcium concentration, you see it's generating some spikes. And then if you look into the interspike intervals here, you see that they actually have kind of a large distribution. They can vary between 110 or something up to close to 300 seconds. A large variability. Same is true for microglia, another brain cell. So also simply put under the microscope, doing some spiking. Again, interspike intervals do vary a lot. So for a stem cell, a PLA cell, so we see that it's getting has a regime that is more regular, but still kind of some fluctuations. And then for HEX cells that are stimulated by carbachol, by a hormone, so then you see that the interspike intervals become rather regular. So meaning that there are different levels of variability in the cells. And to make this more systematic, we can now plot the standard deviation over the mean for each of these cells that we measured, and this is what you can see here. So on the x-axis, it's always the average period, so the average interspike interval, and on the y-axis, you see the standard deviation. So and then for each of these cell types, so one dot corresponds now to one cell, and for each of these cell types, we observe a very strong linear dependency of the standard deviation on the mean period. So and these are also very much on the same scale. So we have here the standard deviation is in the range of 300 as well as the average. So does anybody rings already a bell? So what this smells like? For which process do you expect that the standard deviation is similar to the average? Poisson, exactly. So and how do we get there? So an interspike interval can have this deterministic part that is needed for the channel recovery period and bringing the calcium back into the endoplasmic reticulum. And then the stochastic part that is needed for the nucleation procedure that is then actually similar to a Personian process. For some reason that will become later, so we actually assumed they're a time-dependent Personian process, so meaning that the rate is not constant but has a relaxing term here to consider the refractory period of the channels, meaning we have the activation rate here, the nucleation rate to bring these events together, and then the recovery rate for channel reactivation. So with that one, you can then write down the probability density function in this form here, and we can now calculate and predict the corresponding dependency of the standard deviation over the mean period. And then if the recovery rate is large, so then we're ending up with a pure Personian process. So slope of 1, the sigma equals the average. So then if for slower recoveries, so the slope will decrease. So and this is actually what we also have seen in these experiments. Okay, so far we see good evidence so that actually the stochastic part of calcium spiking is relevant. The other part is so that it should depend also on the spatial coupling between the release sites, because nucleation has to bring together these local events. And how to test that is then in these cells is that we were measuring cells for some time as a reference period, and then we loaded an additional calcium buffer to the cells. What is the calcium buffer now doing? So calcium that is released from one release site is now caught by the buffer and cannot activate the neighboring channel clusters. So and thereby the lambda rate should be decreased. And actually this is actually what we then observed is that faster spiking is turned into slower spiking, so for all cell types, and also the variability, as you can see, is increasing for all of these cell types that we measured. Again, the sigma t average plot, so standard deviation over the mean period. In red, the cells before additional buffer loading, and then these cells are coherently shifted towards longer periods and a larger variability. So very much in line what you would expect from the underlying time-dependent Personian process. Okay, so meaning we have good evidence for the wave nucleation process under there. Any mean remarks? Why didn't you measure it? Would be nice to measure it. So problem is that the cells are so small, so that it's really hard to get the signal directly. So the experiment that I've shown you by Marchant with this line scan were actually done in oocytes, which are really huge cells. So that's why you can nicely visualize that there. For this more physiological or human physiological cells, so the diffusion is so far that the experimental timescale is really hard to grab there. Okay, then this slide I put in last night because I remember that Matthias was making the comment about temperature is such a global variable, and as you can see, it's also true for calcium signaling, where if you measure cells at 20 temperature and then switch to another temperature to 30 degrees, you increase the temperature, you get rid of your calcium spiking. If you switch the temperature back, you re-evoke the calcium spiking, right? So, which is kind of, well, I think for physicists it's fascinating, it's interesting, but it actually had a rather larger implication because when me as a, outsider, so the alien, entered the lab to do these experiments, I was told that astrocytes in slices, in mice slices, are so different compared to astrocytes that you culture in a dish. Because one shows this kind of spiking, the other one do show totally other spiking. Then I was doing this experiment and looking left and right, and I realized, okay, why do they have a heated buffer and there is no heated buffer? And then I was the one who then changed actually the heating there, and then what you can actually then realize that they both follow the same thing. So now if you measure the spike widths, yeah, so of these spikes for cultured astrocytes and as astrocytes in slice, so they all lie on such a exponential curve. So this said, these cells are still different, so they show different expression profiles and so on. But in terms of calcium spiking, so the difference is actually mainly driven by the temperature. Okay, small side note. So how bad am I with time already? Okay, I have to hurry up. Okay, then to demonstrate the things and to understand things better, we then made a multi-scale model, so where we were simulating individual channels by Markov chains. So we then have a flux description for the clusters, and we then were solving the reaction diffusion system analytically by coupled Green's function on this level. And when we do this, so that's why I call it a cubistic modeling strategy, because we have here an analytical solution that determines the states of our channels that do then drive back, drives the dynamics of the reaction diffusion system. If we do that, so then we can actually show that without any feedback on the IP3 dynamics or endoplasmic reticulum, we can generate the whole spectrum of cellular responses. So the upper row is actually measured calcium signals, and then the lower parts are the simulated ones, where it's actually key that this inhibition of the subunits, so in the magenta line, is a driving mechanism for the spiking. So we skip this for time reasons. So now from our simulated measures, so we can then also look into the variability. We see again a slope of 1 for the cells. We can do an in silico buffer experiments, again demonstrating that fast and more regular spiking is turned into slow and irregular spiking due to the change in the spatial coupling. And then we were trying to find out what is the different slopes. Why do different cell types exhibit different slopes? The only thing that we found back then was actually that if you overstimulate, if the flux is so large that you oversaturate the inhibition effect, so that you then actually can decrease the slope, which was not really satisfying. That's why we looked a little bit more into detail. So meaning here you have now astrocyte spontaneous spiking, stimulated hex cells that has a smaller slope in this dependency. Again, from the distribution probability density, we can calculate these moments, and then we can map these different cells on and fit the different parameters, and we see that the stimulated hex cells have this very small recovery rate, so they are in over inhibition compared to the spontaneous spiking in the astrocytes. Now we can do a little bit of information theory there as well. So information as entropy export, and then, well, you haven't brought this up, but it's a relative entropy, right? Also called Kullback-Leibler distance. That is a measure for entropy for information gain, and it's mainly measuring the relative entropy of this distribution compared to a reference distribution. Yeah? Everybody knows that, heard about it. Well, now you did. So in our scenario, we can now plug in our pure Poissonian process, right? So the most random part that we observed, and then to compare that with a part where this recovery rate plays an important role, you can do the math, and actually you see that this all only depends on the ratio between the nucleation, so the coupling, and the recovery rate. So this is a harmonic series, and then you get this expression is only a function of this ratio. And then also the slope is only there, so meaning that the slope is kind of indicating the information content of the environment. So for spontaneous spiking in glia cells, we do not have anything, no information in the environment, and the driven and stimulated cells exhibit an information gain. Okay. I will skip this part because I think some people of you will be more interested in the energetic part. The bottom line here is that we can then do further measurements, and just because this was today in your talk as well. So if you now stimulate cells with two different strengths, so you look for the pattern of life, and what you actually identify is that it is a linear relation between the first spiking period and the second one. So, and it turns out that actually this is independent from the starting point, meaning that you have here something like a Weber law of perception. Yeah? So that meaning, so like chemotaxis can work at a large range, or how we can adapt to light in dark rooms and still see something in the sun, is because we have here kind of a fold change behavior. Okay, so we can then do more and more about this one, finding a thing, but let's talk about energy. So, where we are interested, what does this regulation of calcium triggering mitochondrial activity that is producing the ATP, and the ATP is then needed to pump back the calcium into the endoplasmic reticulum. How does this crosstalk work? So we can start here and write again a mechanistic model to describe this, this scenario, where you have seen all these things already before, except the explicit exchange between the endoplasmic, the cytosol and the mitochondrium and the pumps. So from the model, we can now look what's happening if we have difference in the substrates leading to difference in the ATP resulting ATP concentration, what will happening with the calcium dynamics. And so from that one, we would hypothesize and predict that if we have a high mitochondrial substrate, we have slower oscillation compared to a low substrate or starvation condition. So, and you can quantify that. We then went into the lab and actually found that one. So where for high glucose content in the medium, so the spiking was typically slower compared to starvation conditions, which is actually not 100% true, but only true if we starve the cell for 1 day or for 1.5 days, because before there has been glycogen. Remember glycogen, the molecules as an energy reservoir. Only if cells were then depleted from this buffer mechanism, actually we then could retrieve our prediction also in experiments, so that there has been faster spiking under starvation, and that also the ADP to ATP level then increased. Yeah? Okay, so meaning that this coupling is then giving like a rescue mechanism if ATP goes down. So the pump activity goes down, which brings the base level of in the cytosol up. This is now, so the cells with the experiments has been astrocytes, Hex cells, and HeLa cells. So we did it with 3 different cell lines. Okay, so if ATP goes down, so the pump activity goes down. So meaning then we have an increase in the cytosolic calcium level, which is then increasing the open probability of these channels, right? Remember the positive feedback. So if you have then higher calcium signaling activity, that is then eventually driving mitochondrial activity and thereby compensating the energy demand. Okay, which might be then also the things that we observe in cells when cells do, without any stimulus, exhibits this kind of spiking, may point actually to some housekeeping activity where cells try to keep their energetic balance in a healthy condition. Does this have any implications? So we do this in the context also of Parkinson's disease. Parkinson's disease is actually classified or characterized by mitochondrial dysfunction, so a deficiency. So, and there are Carmen's and Sunda's posters. So Carmen is still coming up, I think. who's working on the LARC2 mutation. So if everything that you want to know, you can ask her then. We have done some first experiments, not with Carmen, but in another thing, so where we were now measuring calcium activity in these cells that carry this mutation and have mitochondrial defect, and compared that to cells that have the same genetic background but do not carry the mutation. And we see that these Parkinson's cells do exhibit faster spiking compared to the wild type. But there was no change in the sigma T average relation, which why I lost a bottle of wine, and I'm still sad about this, and that's why Carmen has to redo the experiments. Okay, then the other question is, so if we now force some calcium signal in the cells, what is happening with the mitochondrial activity? So changing the input signal, so by stimulation, so we see that more stimulation, on average, the cells spike faster, and we can measure the glucose uptake rate in these cells, and we see the opposite trend. So if we then plot that for astrocytes, we see so the slower the cells spike, the lower the glucose uptake rate becomes. The faster the spiking, it's increasing the glucose uptake rate. And the same is true also with some different numbers there for HEX cells and a different stimulation. If we now bring this into the general framework, so we have this encoding relation for calcium signals, and the calcium signals are then leading or controlling the metabolic flux. Can we bring this into some context? If we now look there into this metabolic decoding relation in terms of the calcium frequency in the flux, here for one cell type, here for another cell type, and you see that we also used different substrate, not glucose but glutamine, following the same behavior, similar to the input-output relation that you have seen in Thierry's talk today, right? This kind of sigmoidal saturating, potentially saturating curve, which fits somehow with the activity of this enzyme, of this calcium-regulated enzymes. So in this case, you see it for the alpha-ketoglutarate dehydrogenase, that independent on the calcium concentration, it also exhibits the sigmoidal curve. We then went back into the model and tried to see if we can get similar behavior there. Interestingly, for our deterministic model, so we did not really observe this kind of behavior. So meaning that this model is probably wrong under these assumptions. If we made a stochastic version out of it, so then we could reassemble something that looked kind of sigmoidal again. So, but here I'm hand waving. Okay, I'm just like coming back to all models are wrong, and some can be useful under some circumstances. Okay, so that's an indication that this crosstalk is important, can be meaningful, but what's about the bigger picture, right? So, and then there we teamed up with Francesco, Gian Maria, and Valerie, so where we have now generated a more detailed model of this crosstalk. So you have the TCA cycle here again, right? So you're already used now, you always see only TCA cycle when you look in the slides. The positive regulation of calcium for these two enzymes, and then the crosstalk with the calcium machinery. So here you see that the pumps, so the Zucker pumps pumping out calcium is needing ATP, and the ATP comes from the mitochondria, from the electron transport chain. So now, who can derive the emergent cycles for the system? Okay, then I won't do it here because that would be the whole lecture, I think, to explain that. But obviously, so our output, right, is our ATP. So it's the outer part. So and then we have the intrinsic TCA and the Oxford cycle, and then having so the one cycle becomes evident from there, and then the other two cycles, the input cycles, are then given by the chemostating of the environment, right? So now, having this defined, we can now define the efficiency of mitochondrial metabolism, and what you first can observe here is so the model is reproducing the previous results. So meaning for high substrate, lower oscillations, there's a region where we do not have any spiking at all, but then the spiking occurs on the calcium level as well as in the ATP level. For low substrates, so we have again the faster oscillations. Now putting this as an input into the CRN framework, the interesting thing that we see is now independent on the stimulation strength. So we get then, and I put now the color scheme here, so that was asked Francesco. So the color scheme of these dots corresponds to the frequency. We see that in this part here, so we do not have any calcium spiking. So then the efficiency goes down as well as the ATP concentration in the cytosol goes down. Then at this point here, we have the bifurcation point where now calcium spiking starts. So, and the calcium is now pushing mitochondrial metabolism. So that's why we, first of all, we get a strong jump into the ATP concentration again. Calcium is boom, pushing mitochondrial metabolism. And also the efficiency is getting a boost. And then the faster the spiking becomes, so the efficiency starts to go down, and then at this point the spiking stops again. And then efficiency and ATP concentration follow here more or less the same trend. And so that is now for one regime, but this phenomenon is rather stable and is also then holds for the other regime. Okay, so meaning the takeaway message here is, well, yeah, this crosstalk is, well, not only being a rescue mechanism, so if cells are getting really bad, but it's very likely that it's also a way how cells work in an optimal efficiency regime. So, okay, so far, nice theory, you can tell us everything, so I only believe the math that I cheat myself. But actually, so we have now done the first experiments, and what you see here is now, and again, in astrocytes so far, we give different stimulation strengths that lead to different calcium spiking. And then we see, at least in this one experiment with these 2 replicates, we have now a few other experiments in other cell lines. We see this nonlinear behavior that kind of resembles potentially this nonlinear behavior in the ATP concentration. So that's why I'm very hopeful and a strong believer so that this thing is real. So we are following this up now currently with also stable isotope metabolomics that Matthias didn't introduce, Carol didn't introduce, I won't introduce it neither then. Yes? No, the color represents the frequency of the spiking. So meaning, so it's here we get slow spiking, and then the spiking becomes faster and faster. Because here we look into, wait, go back. Because here it's directly linked to the here it is linked to the stimulation level, yes. So, and then cells do at this point, so cells do start to be fast, yeah? And then they're getting slower and slower. So, but the efficiency, so it's not that you only need to be just spiking. Apparently, you have to have a specific frequency here. So it's not directly at the onset. It's not like a jump, right? So, but you get this region where you have this optimal area. Is that clear? Or why would you expect that you need the red dots here? Okay. Okay, what's that's then I show this Matthias in the private sessions, how spatial arrangement of the cell shape metabolic profiles. But we'll spend briefly 10 minutes on the second part that I promised. that calcium drives cell adaptation, cell differentiation, and thereby nanoevolution. So, and then one way to understand, or one phrase to, or one framework to put it is, so the Weddington landscape, where Weddington made a gimmick, so to say, okay, we have a stem cell that has a high potential, and then during development, it's rolling down this hill and then getting into the right cell type. Yeah? So that's has been the idea, how to explain genetic interactions of development. And here we still do not really understand exactly this force that is driving and determining the cell. So for some parts we do understand, as for fly development, for instance, so Thierry already gave you some hints how this can be understood. So, but again, to have a complete picture, what we are doing now is single cell RNA sequencing, right? So we have now the technology to read the transcriptome of individual cells by so something that is based on droplets, so that's why it's also called, the first version of it was called DropSeq. What you do is that you generate 1 million of droplets per minute, and then you isolate individual cells in this droplet with a bead, and the bead has, well, a sticker to catch mRNAs as well as a unique identifier. And then what's happening in these droplets, you then capture all mRNAs of, or not all, but most mRNAs of a cell together with an identifier. You can put everything into the sequencer, and then you sequence from the one side, you sequence the mRNA, so you know which gene it corresponds to, and from the other end, you get the identifier, meaning that in the end, we assemble for 16,000 genes, the expression of 16,000 genes, or up to 18,000 genes for thousands of cells. So, and that's now really a way how we can try to understand development of, well, cell lines. It's a more boring one, but also for more complex organisms. That would have been your prize. We do that in the context, or what I will show you is that we do that in the context of epithelial mesenchymal transition. So there has been some cancer people, right? So I've seen 2 posters there. So what epithelial to mesenchymal transition is doing is that it's changing an epithelial cell type, so which is the tissue cell type, into a cell type, into a mesenchymal cell, and these mesenchymal cells have more stemness character, and they can crawl, they can crawl, and thereby spreading the message. It's important in development, where obviously we have to build up the tissue, right? So that's why cells have to move at the right location. It's important for wound healing, so where we have to generate new tissue, so like Carol with her sting at her arm right now. And the dark side is in cancer, where if you have a tumor that is made of epithelial cells, so these cells can then switch into a mesenchymal cell that has higher stemness character, can crawl, and is thereby one mechanism how metastasis is happening, right? You have a tumor cell that starts to crawl away, entering the bloodstream, traveling somewhere, and can spread the tumor at a different location. So we use their cell line, so a Hamler cell line that is a model system for breast cancer, and they have intrinsically these 2 different cell types, and they can switch between each other. So here you see a microscopy picture, and then there are cell surface markers by which you can characterize them. So these are the mesenchymal cells, and if you now take only these mesenchymal cells out and then replate them, you see that they will generate again a heterogeneous population. So therefore, this cell line has intrinsically the property for phenotypic switch. And we do that now more systematically, so where we force cells to have a specific calcium signal, and then we read out the transcriptome and see how different calcium signals are changing, where we have a perfusion system that is shown here. And you see that now, can you see it? So there's a synchronous blinking of cells, okay. And we can now force different calcium signals in these cells. So that is this set of experiments here where we can go with different periods, but we also then can trigger them in a stochastic pattern. So we can then also or use cells where we just gave a constant external stimulus, so that cells do spike in the eigenfrequency, and we compare that to different controls where there's no stimulus, no forcing, so meaning like the resting state. Okay, what can we then do? So first of all, here you see the typical U-map representation, so which is a dimensionality reduction. Each dot corresponds here now to a cell in a projection of this gene space, 16,000 dimension, into a two-dimensional map here. And you see so that we can distinguish the two different cell types and that the different treatments do populate both populations, right? So we see, so there are mesenchymal cells, if we treat them with different spiking or constant treatment, they are always there, as well as for the epithelial part. We can look then there into the cell cycle gene, so just for the cancer people, that's it's kind of important, but the more interesting part is then that we can now also look into gene sets that corresponds to this transition. And then we actually see that, so this EMT score, so the epithelial mesenchymal transition score, is more prominent at these tips, so meaning that these corresponds now to cells that do the phenotypic switch and are therefore driven into the different cell type. Okay, everybody knows what the differentially expressed gene is? Okay, so in brief, differentially expressed gene is a gene that is different in one condition than to another condition. Yeah? So in the end, so here you see now the expression for a specific gene, so also important in T cells. Each dot corresponds now to the expression level of a specific cell. And then in the end, what you want to know, so is this gene in this cell type different compared to that cell type? In the end, it's comparing two distributions. Yeah? So sounds easy. So as you can see, this data is rather noisy. You have a lot of genes, so meaning that this is an own game, so where you have to develop your own statistics so that you believe it, kind of. Anyway, so meaning DEG, differentially expressed gene, just means so that the gene is different under specific conditions, and we map this now onto our different signatures that we have, right? So meaning that we can compare now a constant ATP pulse, so where we have a permanent stimulation of the cell, to a control condition. So and then we see so that under this condition, so we have observed about 1,500 genes, so where cells lay around here. So if we now look into forced oscillations, yeah, so we force them with either with 1 minute, 2 minutes, or 6 minutes. So we see actually, first of all, that the number of differentially expressed gene is not or is smaller actually for the slow oscillations than for the controlled stimulus, and is increasing the faster the calcium becomes. Yeah? The faster the calcium spiking becomes, so we have kind of a dose response curve in number of changed genes. And the interesting part is so that now for a stochastic pattern, yeah, so it has now this has the same average period as this guy here, but in a stochastic pattern. And then the number of differentially expressed genes is highly increased. Yeah? So meaning that this diversification is leading, so diverse input signal also leads to a much more diverse gene expression profile, which is not trivial, because classically, so you would consider it's kind of an integrating system, so that the calcium is integrated in the nucleus to generate the corresponding gene profile, but apparently it's not the average, but it's actually the concrete signal that is driving the gene expression profile. This depends a little bit on which cell types you're using. So the main difference in this breast cancer cell line was actually linked to inflammation. So inflammatory genes. So which are and some apoptotic genes, probably because you have here also faster calcium spikes that come directly to each other, which is then a little bit linked more to this 1 minute part. So, but we are still digesting this complex data set. Another thing that we can do, and then this is the final slide, I think, I hope, more or less, yes, has to be, is that we're looking only for the constant stimulation level. So we stimulated cell with either 100, 50 or 20 micromole, and we were now looking into the dysregulated genes in the epithelial. This thing is in the epithelial cells and in the mesenchymal cells, and we split this into genes that are downregulated and upregulated, and again in downregulated and upregulated. And the thing that I still do not understand, and I'm looking forward that you will solve the problem in the next 2 days for me, is so that it's cumulative, right? So meaning, so genes that are dysregulated for a 20 micromole stimulation are also dysregulated with 50 and with 100 micromole stimulation, and then you add additional gene sets that are dysregulated, but the majority of genes stays the same. I think this is, I do not have an explanation for it, so why and how, but this is rather robust. So we have now this data set, another data set, we use different control versions to it, and it always stays the same. The message is so that there is kind of a cumulative fold response. Okay. With this one, I think I end with a final movie to make then also commercials for the posters of Parisa and Najme. Was good, Parisa? Yes. And what they are doing is that they use zebrafish to understand brain behavior, also in the context of epilepsy. And what you see here in this movie is actually now a zoom into a zebrafish larva brain. And you see the activity based on calcium. So, and if the movie starts now again, so you see these are the eyes, and then each of these dots corresponds now to a cell, and you see how a seizure, an epileptic seizure, is bubbling up and running through the brain. And why does the seizure stops? Typically, why do epileptic seizures stop? Energy deprivation, right? So that's typically seizures end when the cells are out of energy to refill their iron gradients. Okay, so then preaching to the priests, life requirements, you know them, live them. Who recognized that one person that was not before on the slides on this upper row? McClintock, yes. So because I think that's really important, maybe the most important message that I want to give to you, so that you should also have a feeling for the organism, right? So for all of us who invading biology and try to make sense out of life, we should not forget that it's not only physics. At least it's also biochemistry. Okay, with this one, I say thank you. So also for staying, please stay curious and try to decipher the patterns of life. So when I was walking this morning to the beach, so probably you do not see it, but I think I found evidence for applied path integral things, because these ants were walking along this border here and then making a perfect turn to the nest here. So it looked really like optimizing the path in terms of efficiency to bring things home. The bad part was then, coming back to Anupam's replicate thing, so when I went back later, so the pattern was gone. So that's why I do replicates. Okay. why it's the optimal path. So, well, you don't see it, but the street was really long, so it was for at least for 10 meters. They were walking really at the border of the street. So, and then only when they had to ended up here. So then they only at the last part of the street they took the turn through the hilly and not so easy environment. But probably just random. I agree. So replicate didn't work. So okay, thank you.